Hey guys, and welcome back. Thank you so much for joining us again in our biology series. Today we get to talk about cell division, so mitosis, and also some cell differentiation. So pretty cool stuff, guys. Make sure to pause, rewind, watch as many times as necessary so you can become a pro at this. All right, let's do it. Science rocks.
Like you know by now, it is super important for you to create a biology notebook. So open up your Google Drive or Spiral Notebook and take some notes. Go ahead and copy down these 23 vocab words. Remember, they go in ABC order like this. And just come back later and create your own definitions of these words. Remember to use your own words. Drawing and labeling the phases of mitosis would be a for sure thing you would want to do as well. So go ahead and pause this if you need to finish writing down the terms. Cell differentiation. So, multicellular organisms possess many different types of cells. These different cell types carry out specialized functions that are necessary in order for the organism to survive. The process by which a cell becomes specialized is known as cell differentiation. Offspring are formed when a sperm cell unites with an egg cell during fertilization. Okay, here's the sperm cell, here's the egg cell. So during fertilization, an offspring is formed. The fertilized egg, or also known as a zygote, then undergoes several cycles of cell division until a large ball of identical cells is produced. Okay, cell division, then you've got a large ball of identical cells that's produced. So eventually, this large ball of identical cells undergoes cell differentiation. It can also be called cell specialization. Okay, so during this process, cells become specialized to perform different functions. Okay, maybe they're going to be a bone cell or a red blood cell or a skin cell or a muscle cell. Okay, the cell cycle. This is something important that you'll want to draw and label and put in your notes. So all new cells come from pre-existing cells. All new cells come from pre-existing cells. The cell cycle refers to the series of processes involved in the growth, development, and reproduction of a typical eukaryotic cell. The two main phases of the cell cycle are interphase, okay, it's going to start here, interphase, okay, and the M phase, okay, this is the M phase. And then, of course, you have cytokinesis, where the cell finally pinches into two. So, most of a cell's life is spent in, look at the cycle, most of the cell's life is spent in interphase. This stage includes all normal cellular functions associated with metabolism and growth, okay? So DNA replication, the duplication of a cell's chromosomes, all right, occurs during a part of interphase called the S phase, okay, called the S phase. So at first the cell is going to grow. It's going to enter the S phase where its DNA is replicated. DNA replication. All right, that's important because eventually when the cell splits into two during cytokinesis, you want both cells to know what to do. So they're going to DNA that, they're going to replicate that DNA so this each cell will know what to do. Okay, the cell division begins in the M phase. Okay, after it grows and after it replicates its DNA in the S phase, and after it prepares for mitosis in the G2 phase, the M phase is going to include mitosis, all right? So during mitosis, the nucleus of a cell actually divides into two daughter nuclei that each contain the same number of chromosomes as the parent nucleus because remember, the DNA replicated. All right. Following mitosis, the two nuclei are separated into two identical daughter cells during cytokinesis. Cytokinesis is, here's the parent cell, mitosis happens, you've got all these steps, and they finally pinch into two during cytokinesis. Okay, the cytoplasm divides and the two new cells are formed. Here's an example of an animal cell, okay, and here's an example of a plant cell. So here's a step-by-step of the major events that occur during mitosis, and then it ends with cytokinesis. It actually pictures interphase up here. So it starts with interphase, goes through mitosis, and it's going to end with cytokinesis. This is something that you'll want to kind of sketch out, draw, label in your notes. All right, so this first one up here, I'm just going to put an I for interphase, okay? So this first cell represents interphase. Let's see, a cell spends about 90% of its lifespan in interphase. Remember the cell cycle, and it showed that interphase took a long time. During interphase, remember the cell grows, and it will eventually duplicate its genetic material, that was the DNA replication, in preparation for mitosis. Okay, so when that's all done, it looks just like a scrambled mess here. So when that's all done, grown and duplicated and prepared for mitosis, it's going to have the first step of mitosis, which is called prophase. Okay, the first step, I'm just going to put a P for prophase. All right, during prophase, the genetic material, which is the chromatin of a cell, condenses, okay, into rod-like structures called chromosomes. So notice an interphase, okay, it's all a wiggly mess, and in prophase, they condense into these rod-like structures called chromosomes. So if I were to take this red chromosome right here and make it a little bit bigger to show you, all right, this would be one chromosome, okay, and because the cell contains double its normal amount of genetic material, because remember in interphase it doubled its DNA, each chromosome, okay, this is a chromosome, each chromosome is actually comprised of something called sister chromatids. Sister chromatids. This side is a sister chromatid and this side is a sister chromatid. So each chromosome is comprised of two sister chromatids, and they're held together in the center right here by something called a centromere. Think center, centromere. Okay, so once again, I've just taken one of these red chromosomes and drawn a bigger picture of it, okay? Notice in prophase, there's this dotted line, this dotted nuclear membrane. In this next phase, it's gone, okay? So the nuclear membrane that is holding these chromosomes all together is going to dissolve, all right? And then we're going to move on to our next phase of mitosis, which is called metaphase. Okay, this cell right here is representing metaphase. The microtubules, these little dots on both ends of the cell, are going to attach to the centromere. Remember, the centromere is in the middle. They're going to attach to the centromere of each chromosome, all right? And they are lined up in the middle during metaphase, where they're attached by these microtubules that are going to help pull them apart here in a little bit. So think meta, middle, okay? The next phase is called anaphase. I'm going to put an A for anaphase. The chromosomes are pulled in opposite directions, all right? They're going to be pulled in opposite directions. So they start to separate, okay? They are pulled to opposite sides of the cell. So notice one sister chromatid goes this way and the other side of this red one goes this way. So during anaphase, they're being pulled apart. All right. The next phase is called telophase. And during telophase, a new nuclear membrane forms around each set of chromosomes. All right. Notice this dotted nuclear membrane. All right, and you've got the sister chromatid over here and one over here, and you've got their nuclear membrane keeping them together on both sides. Okay, that's telophase. All right, both sets of chromosomes begin to decondense, all right, back into the chromatin, okay, the squiggly mess I was talking about. All right, so after mitosis, And these phases right here are mitosis. Okay, so we'll label that out. Mitosis. And to remember it, I have it over here, PMAT. PMAT means really nothing on its own. I think of a puppy in a PMAT. Okay, so puppies in a PMAT. It just helps me remember the order of mitosis. Okay, mitosis, PMAT. So maybe PMAT will help you remember the order of mitosis. So after mitosis is cytokinesis. I'm just going to put a C for cytokinesis. So during cytokinesis, the cytoplasm and the organelles are divided, okay? They're finely divided and they're pinched into two. So two daughter cells are formed, okay? In cells that lack a cell wall, the cell pinches in two. And in cells that have a cell wall, like a plant cell, something called a cell plate forms between the two new cells. Make sure to take a look at this video as well sometime, explaining more about mitosis. All right, let's do some practice questions. Number 1, examine the graph below. According to the graph, what happens once cells reach a certain time? Okay, so it's got time on the x-axis. I know this, so independent variable. And it has cell size on the y-axis, your dependent variable. Okay, what you're trying to figure out, what you're trying to measure. So what happens once cells reach a certain time? Okay, so look this graph over, look at your answer choices, and see what you think. So organisms can grow when there is an increase in their number of cells. All right, the number of cells in an organism can change as a result of cell division, like we just talked about. So cell division is often triggered when a cell becomes too large. All right, once a cell reaches a specific size, it divides into two smaller cells. All right, and then the process begins again. Okay, so if you're looking at number 1, which one should you pick? A, the cell divides to become two smaller cells. Okay. B, the cells fuse to become one large cell. No. C, the cell dies and two new cells spontaneously appear. No. D, the cell releases fluid so it can shrink in size. No. All right, so let's go with A. The cell divides to become two smaller cells. Number 2, a bat-eared fox has 30 chromosomes in its body cells. When the fox's body cells divide by mitosis, how many chromosomes will each daughter cell have? Okay, so you've got the body cell with 30 chromosomes. All right, so when mitosis happens and you end up with two daughter cells, how many chromosomes are in each daughter cell? All right, so pause this and see what you think. All right, so body cells undergo a type of cell division called mitosis, like we just talked about. And during mitosis, the daughter cells produced are identical to the parent cell. Okay, so daughter cells resulting from mitosis in this bat-eared fox would also have how many chromosomes? 30. Each daughter cell is also going to have 30 chromosomes. Number 3, what is the name of the process that appears in this diagram below? And I like this diagram because it's written out horizontally instead of vertically, so it's, the same thing. And if you have paid attention at all today, then you would probably guess your way through that this is talking about mitosis, right? It's going to start off with one cell, undergoes cell division, and end up with two cells that are identical to the original cell. Number 4, before a cell enters mitosis, what happens to the genetic information in that cell? All right, so pause this, think it over, think of the cell cycle. All right, so if we're thinking of the cell cycle, and maybe draw that out here, okay, remember this first phase. Say we start here, this first long phase happens, okay, before it gets to this next phase here. And then lastly, it's going to pinch into two in cytokinesis. So this long phase called interphase, remember of the cell cycle, interphase. There were actually three different parts of interphase. All right, there was that G1 phase, there was the S phase, and there was that G2 phase. Okay, so if you remember something about the S phase, something was synthesized or made, remember that DNA was duplicated, DNA was replicated, it was synthesized. Okay, so before a cell can even enter mitosis, this phase here is the PMAT of mitosis, and here we have cytokinesis, and right here is the long interphase phase. So before a cell can even enter mitosis, what happens to the genetic information? What happens to the DNA? Remember, that information, that DNA is duplicated, it's replicated. Number 5, most multicellular organisms are composed of cells with different shapes and sizes, and those cells perform different functions. So, what is the best explanation for this? Remember, we talked about cell differentiation. Okay, we're going to look at A, environmental conditions influence gene expression, which determines how a cell develops. Okay, and let's go through the other ones just to check this. As cells divide, we're at B, as cells divide, they only pass on genetic material that will be used in daughter cells. False. Okay, they're going to pass on everything. Let's look on at C. The structure and function of a organism's cells is determined by the genetic material it receives during fertilization. Okay, so remember, fertilization happens, and then all that cell division happens, and you get that big group of all those same cells. So it's not straight from that information from fertilization. And then let's look at D. Mutations accumulate in the organism's cells, causing them to become different from the other cells, okay? Mutations can happen, but not all the time. So we are going to cross off D. So let's read A again. Environmental conditions influence gene expression, which determines how a cell develops. Last one, number 6. The stages of mitosis are shown below, but they are not in the correct order. Okay? Arrange the stages in the order they occur. Okay? So we've got 4 different pictures. Okay? And we need mitosis to go in the order of PMAT. Okay? Remember, PMAT is prophase, metaphase, anaphase, telophase. Okay, so prophase would be 1, metaphase would be 2, anaphase would be 3, and telophase would be 4. So let's look at these pictures and just number them out. All right, first let's look for prophase. All right, we're looking for prophase. I'm going to look at this one over here, number 1, where it shows the chromosomes. All right, and they are 2 sister chromatids bound together by the centromere. For metaphase, number 2, we're going to go with this one. You can see that they are lined up now in the middle. Remember, middle. For number 3, for anaphase, is when they start to separate and pull apart to opposite sides. And then number 4, for telophase, is when they start to look like 2 daughter cells, and then eventually the cytoplasm will pinch into 2 during cytokinesis. So we're going to answer this with 4, 3, 2, and 1. So, after fully covering the topic of cell division and differentiation, you should be able to use models to illustrate that in multicellular organisms, individual cells divide to form 2 identical cells. all multicellular organisms begin as a single cell, which undergoes this process of dividing. Cells undergo mitosis in order to grow, repair, or replace injured or worn out cells. So, I challenge you to create a project, a presentation, an experiment, a model of the cell cycle or mitosis, and show your teacher that you can explain all of this.